Okay, almost done here. Let's see. This is the wrong. This is wrong. Wrong. Okay. Sorry. Okay. What you see here in this diagram, all these striations, these are called red beds because they're red. And what this signifies is iron oxides that formed when the Earth first started producing oxygen. Around 2.5, 3 billion years ago, and all that oxygen got absorbed by the iron in the layers of rock in the earth and the ocean, and basically the oxygen levels did not increase for a long time until all these red beds were filled up with oxygen, and then it could start to build up in the atmosphere. This is a beautiful example of this. This is in Algeria. I forget the exact name of this, but that's in North Africa. Because we're IB and we gotta know about the world. Okay? But the atmosphere, photosynthesis changed everything. It changed everything. Before photosynthesis, the atmosphere is based on carbon dioxide, some methane, nitrogen, but now it's going to have to be based on oxygen. If you couldn't deal with oxygen, you're going to have problems. So here's a kind of graph that kind of shows, the beginning of the oxygen 2.5 billion years ago, and then the gradual increase. Stage 2, this is with the red beds here. You didn't really see much increase. And then it starts to come out of the ocean, but still absorbed. And eventually it fills up. The sinks, it says ocean sinks, and remember, any kind of a sink we talk about in geological terms, that's storage of something, either energy, oxygen, carbon dioxide, or carbon sinks now are big because, used to be a lot of carbon dioxide held in the ocean and on land and trees, but now frozen in rocks, but now those are thawing out, the ocean's getting warmer, trees are being removed, that those sinks are leaving, and that there's more CO2 entering the atmosphere. And you notice here, this kind of interesting point. You see here, there were higher levels. This was about 400 million years ago, and this is what the time of the Earth we had giant insects, because insects can't get too big now because they don't have a circulatory system. But back here, there was so much oxygen. It was so dense in the atmosphere, they could actually be bigger and get enough oxygen. You had huge flies this big flying around. Well, not that big, but they were bigger, which is kind of freaky to think about. So this is the absorption spectrum showing you, remember, photosynthesis likes blue and red. This here in the green, yellow range, not so well appreciated. So here is like the action spectrum, absorption spectrum gets kind of confusing, but the action is this is the action where you do see working of photosynthesis. Remember, chlorophyll likes here these two peaks, 740 looks like, and 400. But there are other pigments that can absorb in other ranges. But so that's why the action spectrum is a little wider than the absorption from chlorophyll. So you got other pigments filling in the pieces so you can be more efficient with the sunlight. But primarily the action happens around the violet, blue, and the orange-red ranges. All right? Now you need to draw this on this page or somewhere. You need to draw that, and you're going to upload it to me. So you have to be able to design experiments. And gosh, I don't know if we're going to get to this because it's our last week. So I guess not. There's 2 more labs we need to do. We'll see. But we already looked at rates a lot with the catalase. Change over time. Everything is over time. We looked at change in mass, pH. So we're pretty good. But the best way to measure oxygen or photosynthesis is to measure oxygen production or how much CO2. Remember, for photosynthesis, CO2 goes in, oxygen comes out. So if you can measure how much oxygen comes out, that's a measurement of photosynthesis. If you can measure how much CO2 is used, that's the way to do it too. So for example, what we have are these sensors where we can put, say, in a chamber we can put a plant. Say we'll put some leaves. This is my version of leaves. So we'll put leaves in a bottle. We'll stick the sensor right there. That's my terrible drawing of a sensor. Hook it up to a Chromebook. Ooh, yeah, baby. And what we can do is measure the amount of oxygen produced. And what we can do is change different things. We can either, if these plants are in water, aqueous, aquatic plants, we can change the pH of that water, see how that affects it. We can also change the temperature of that water, see how that affects CO2. So if we change the temperature, we want to make sure everything else is the same, meaning we use the same size leaf, the same amount of water, the same chamber, everything else the same. The only thing that changes is the temperature. The other thing we could do, instead of an O2 sensor, we could use a CO2 sensor as well, because we have those. And remember, in this case, oxygen levels go up. If we use a CO2 sensor, those oxygen levels will go down. So we can measure those two things. We can measure CO2 use or oxygen production. And remember, everything else needs to be controlled, kept the same. So the time that we run the experiment, the materials, meaning the bottle needs to be the same. The type of plant we put in there. The freshness of the plant. Don't want to use nice fresh plants in one and then crusty, dusty plants in the other. The temperature that we run the experiment should be the same for each one. So we need to control the variables tightly. That's what I mentioned in that first one. That'd be your independent. Our independent variable would be changing the temperature. Independent variable. And the oxygen produced would be our dependent variable. And over here, these would be our controlled variables. Okay, everything that needs to be exactly the same. So the only thing that is changing is the temperature, if that's what we're doing. Or the only thing that's changing is the pH. Sometimes kids, students will, adults for that matter, will design something. Well, we'll change the temperature and the pH at once and see that. Well, if you do that, it's going to be really hard to see what the results are because you're not going to be able to unmix it from everything else. So that's how you design an experiment. There's also examples in your book. This is kind of a fun lab when it works correctly, but it's the separation of pigments by chromatography. So what you have here is filter paper. You see how it's suspended above the bottom. You don't want to hit the bottom. Down here is chromatography. Chromatography solvent. What it is about 90% petroleum ether, which is like gasoline pretty much, and then about 10% acetone, which is like nail polish remover. Okay, so it's some funky stuff. You can get, usually you want to cover this up, see how it's covered, because it will make you kind of, your head spin. And what you do is, it's not shown very well on this, but you put a little spot of concentrated pigment right there. Little spot. What's going on right now? Put a little bit of pigment here. Now I'm making it bigger. A little spot. Concentrate it. So make a really dark dot of pigment there, usually. And then you drop it into your chamber, and what's going to happen is the ether is going to travel up, capillary action, remember, up the paper to a certain extent, and then it's going to stop. Okay, and what you have to do, the most important thing is Dang it, don't do that. You draw a line at the solvent front. We'll say the line was right here. Wherever that solvent stopped, you need to draw a line. And you can see, come on, there we go. It separates what used to be one pigment. I should have done this. Let me try this. Should have done green. I'm trying to change it. Yeah. So this is our single green dot. I'm making it way bigger than it should be. And you see it's moved. It's not green down here anymore. It has moved up. Go back to red. There. So there's no pigment now. That pigment has been transported. You see it's turned into 1, 2, 3, 4 different pigments. And what you got to do, normally you have a little line drawn right there, which lines up nicely with that. You got to measure out how far it went, which is, looks like it's 89 millimeters. And then you got to measure how far everything else traveled. So this carotene traveled 82 millimeters. The xanthophyll, 39. Is that really? Yeah, 82, 39. Chlorophyll A is 19 millimeters, and chlorophyll B is 11. If you notice, the further it goes, it means it's smaller. If it doesn't go very far, it means it's bigger. So this is a good way to separate by size, which we'll be doing next year with some DNA stuff. And then what you do is you divide this number 82 by 89, 39 by 89, 19 by 89, and 11 by 89. And that gives you what's called the RF value. So let me do that. Got my calculator right here. Why don't you do it with me? 82 divided by 89. I get 92.92, and 39 divided by 89. Remember, you're always using that 89. This gives me 43.43, which is about right. It's about half, right? And chlorophyll A, 19 divided by 89, 0.21, and chlorophyll B, 11 divided by 89, 0.12. So what you need to do, if we did this, we would compare what our values were to the accepted value. I believe that these are accepted. I'll double check, but that's what I remember. So here it's pretty close. We're a little further away with xanthophyll, 0.43 versus 0.71. Really far off with chlorophyll A, 0.21 and 0.65, and again pretty far off with chlorophyll B. But that's the basic way you do that experiment. There's other videos you can watch, but that's how most of them go. Okay. All right, we're done.